Good morning. In this session, we are going to discuss certain functions of the loop of Henle and collecting ducts that is related to osmosis. And then we are going to start talking about disorders of the urinary system or diseases that might affect the urinary system. Concerning the loop of Henle, I would first like to start by reminding you of the structure of the loop of Henle and where exactly it is located. So we have the Bowman's capsule, which is the first part of the nephron, and then we have the PCT, or the proximal convoluted tubule, and then we have the loop of Henle, and then the distal convoluted tubule, and then the collecting duct, which is not a part of the nephron, but actually the collecting duct collects urine from several nephrons, and that's why we would have several distal convoluted tubules that open into the same collecting duct, into the same collecting duct. Now, if we try to remember the parts of the renal tissue, we would find that the outer third of the renal tissue is the renal cortex, whereas the lower inner, or the inner, actually 2/3 represent the renal medulla. So actually, what are the parts of the nephron that lie in the renal cortex? Number one, we have Bowman's capsule. Number two, we have the PCT, or the proximal convoluted tubule. Number three, we have the DCT, or the distal convoluted tubule. And finally, this is CD, we have the collecting duct, but the collecting duct is not a part of the nephron, but only the uppermost part of the collecting duct lies in the renal cortex, whereas most of the loop of Henle lies in the renal medulla, in the inner 2/3, just like most of the collecting duct lies in the renal medulla. So here we have the loop of Henle, like 90% of the loop of Henle lies in the inner 2/3, which are the renal medulla. Now, when the filtrate passes in the loop of Henle, it passes first downward and then upward. So the renal filtrate will first pass downward in this direction, and then will make a U-turn and pass upward to go into, or to reach, the distal convoluted tubule. This is the distal convoluted tubule, and this is the proximal convoluted tubule, and that is Bowman's capsule. This side A, or arm A, of the loop of Henle, where the renal filtrate, which is not yet urine, will pass downwards, which is branch A or arm A, is called the descending limb of the loop of Henle, or the descending arm of the loop of Henle, whereas the other side, which is B, will be called the ascending limb of the loop of Henle. Now, those two limbs, limb A and limb B, are very close to each other. They run parallel to each other, and the fluid in them passes in two opposite directions. That's why the loop of Henle is called a countercurrent system. Countercurrent systems are not only present in the human body, and the best example being the loop of Henle, but they're also present in certain systems, mechanical systems outside the body. And the main function of having such a countercurrent system is to maximize the function. Whatever the function is, the counter current system will maximize the function. If we apply this to the loop of Henle, we will first erase all what's on the screen and draw the two arms of the loop of Henle. Actually, the loop of Henle is U-shaped. So we have fluid moving downward in arm A, and we have fluid moving upward in arm B. And the two arms are parallel to each other and close to each other. So these are the characteristics. So the two limbs are parallel to each other, they're very close to each other, and the fluid inside them runs in opposite direction. Whenever you have this, you maximize the function. So what exactly is the function of the loop of Henle? The function of the loop of Henle is to reabsorb sodium chloride, which is salt, and therefore to reabsorb water by osmosis secondary to the absorption of sodium chloride. So actually, it will concentrate the urine, because when you withdraw salt and then withdraw water, then you are minimizing the volume of water in urine. You are concentrating urine. You might be asking yourself, I am reabsorbing as solute particles, actually. And meanwhile, I'm reabsorbing solvent, which is water. Then the concentration should remain the same. Yes, this is a very smart question. But actually, we are reabsorbing water or the solute, the liquid part, more than we are reabsorbing sodium. So if you reabsorb 10% of the sodium chloride present in the filtrate, you will reabsorb 50% of the water present in the filtrate secondary to the absorption of sodium chloride, and therefore you reabsorb fluid more than solute, so you concentrate urine. So the main function of the loop of Henle is to concentrate urine. And how does it concentrate the filtrate? Actually, it concentrates the filtrate. How does it How does it concentrate the filtrate? It concentrates the filtrate. Actually, this should be filtrate. It concentrates the filtrate by reabsorbing more water, than salt. More water than salt. Again, the main function of the loop of Henle is to concentrate the renal filtrate that reaches it via the PCT. And how does it? So this is the function. This is the main function. And how does this occur? By reabsorbing more water than salt. How is sodium chloride reabsorbed in the loop of Henle? It's not written on the text. I removed it from the content in order to make your life easier. But if you are interested to know, so sodium chloride in the loop of Henle is reabsorbed by active transport, whereas water, of course, would be reabsorbed by osmosis. So what's the relation between this and the loop of Henle being a countercurrent system? I would like to emphasize that the loop of Henle would concentrate urine whether it were a countercurrent system or not. But by being a countercurrent system, the fact that the loop of Henle is a countercurrent system will help maximize the function of the loop of Henle, meaning that it will maximize the concentration of the renal filtrate or urine. This is all what you have to know about the loop of Henle, and now we would move on to the collecting ducts. The collecting ducts are not part of the nephron. This is the first thing we have to know. So they are not a part of the nephron. This is very important to know. Not a part of the nephron. The second piece of information that you have to know about the collecting ducts, that they do not collect or receive actually we do not say receive, because they do not receive from one nephron. We had better say they collect, because when you say collect, you remind yourself that it takes from many nephrons. So the collecting ducts collect urine actually from many nephrons, And although we call it urine, yet there is some adjustment that will be done to that urine, meaning that the filter that comes out of the distal convoluted tubule of the nephron is called urine, but yet there would be some more adjustment that occurs in the collecting duct. So the third piece of information that you have to know about the collecting duct, other than it's not a part of the nephron, other than not being a part of the nephron, and other than collecting urine from many nephrons, is that it is the site where the final tuning of urine concentration occurs, where final tuning or adjustment of urine concentration occurs. So final tuning of what? Final tuning of urine concentration, okay? Since I say that it is a site where the final adjustment or tuning of the urine concentration occurs, how does this final tuning occurs? How does this final tuning occur? It occurs via absorption of water, and therefore the collecting ducts carry out their function, their main function, which is the final adjustment of urine concentration by reabsorption of water. So their main function is by reabsorption of water. And of course, since I say that their main function is final adjustment, adjustment doesn't mean that every time I will reabsorb a constant volume of water, but this volume of water, which is reabsorbed, is variable according to many things, variable according to your water intake, variable according to the weather and the temperature of the environment around you. And that's why we say final tuning. So if you drink a lot of water, then the collecting ducts would minimize reabsorption of water to excrete that additional volume of water that you ingested. But if you drink very little water, then the collecting ducts will reabsorb as much water as they can in order to preserve your body water. And because the collecting ducts are doing that smart function, they are not doing it on their own, but they are controlled by a hormone, and that hormone is called the antidiuretic hormone, and that hormone is secreted by the pituitary gland. The pituitary gland is a small gland, but very important gland that is connected to the brain. And the pituitary gland is called the master gland in the body. This refers to the great importance of the pituitary gland, and it does this, or it carries out this function of being the master gland in the human body by secreting hormones that control many functions in the body. Amongst these hormones is the antidiuretic hormone, and it is secreted by the posterior part of the gland, because this master gland that is connected to the brain consists of two lobes. I'm magnifying it, and the size is out of proportion in order to show you that we have an anterior lobe and a posterior lobe. The anterior lobe, of course, will be towards the anterior part or the front part of the body, and the posterior lobe will be towards the posterior part of the lobe. What does the antidiuretic hormone do? It promotes water reabsorption from the collecting ducts. But this reabsorption that occurs under the effect of the antidiuretic hormone does not occur by simple osmosis or osmosis that does not require any channel, does not occur by the mere passive of water down concentration gradient, but it occurs through specific water channels. These water channels are called aquaporins. So these specific channels are called aqua, means water, and porins, of course, means passages or channels. So what are aquaporins? We are going to see them on the next slide. Aquaporins are present in many cell membranes in the human body, but they are especially abundant in the collecting ducts. And they are there in order to conserve water. So you might be asking yourself, if this is the collecting duct, if this is the collecting duct, and this is the blood vessel, isn't water capable of passing by regular osmosis from the inside of the collecting duct to the blood vessel? Isn't the collecting duct being able of reabsorbing water simply by osmosis from the collecting duct to the blood vessel? Yes, it is. But when there are aquaporins, when there are these protein channels that lie in the cell membrane and that allow the passage of water, they will allow passage of water by osmosis, but this is not regular osmosis. This is facilitated osmosis, because we know that whenever we have a passive type of transport, and that does not require energy, but that passive type of transport requires a protein channel, then this passive transport will be a facilitated diffusion. And here it would not be facilitated diffusion, but facilitated osmosis. We do not use the term facilitated osmosis, but I'm just saying it in order to remind you that when water passes via aquaporins, this is not regular osmosis, but this is a bulky type of osmosis that allows the passage of much more amounts of water, but still passively, without the use of energy, down concentration gradient. So actually, aquaporins facilitate osmosis. Aquaporins facilitate osmosis. So if you want to summarize the information about aquaporins, they are protein channels, and these protein channels, as usual, are in the cell membrane, and these aquaporins facilitate osmosis. How do they facilitate osmosis? They speed up osmosis. They increase the volume of water that is being reabsorbed. They increase the volume of water that is being reabsorbed, and therefore we can just say that they increase the rate of reabsorption. However, this reabsorption is still passive. This reabsorption still does not require any kind of energy. This reabsorption still occurs down concentration gradient. When do aquaporins open and become active? When you want to reabsorb water, when you want to return water to blood, when you want to preserve water. Of course, aquaporins are important for conservation of water, but if you conserve water inside your human body, then they will lead to very concentrated urine. When it comes to urine will, or to the characteristics of urine, it will be, if all aquaporins are open in the collecting ducts, the urine will be very concentrated urine. And since aquaporins are under the effect of anti-diuretic hormone, this makes sense. Why does it make sense? First of all, antidiuretic hormone, whose acronym is ADH. Why does it make sense? Because in medicine and in biology, diuresis means increased urine formation and increased urine volume. So there is increased urine volume and therefore increased urine formation, actually. So when the hormone, when do you increase urine formation? When you have plenty of water inside the body and you want to get rid of them, but antidiuretic hormone prevents diuresis, prevents increased urine formation. And that makes sense because the antidiuretic hormone promotes aquaporins to open and promotes reabsorption of water. So if you want to summarize all this, we would open a new screen and we would write the information that is related to the antidiuretic hormone. So the antidiuretic hormone does what? It opens aquaporins, especially the aquaporins that are present in the collecting ducts, and therefore it helps water reabsorption, meaning returning water to blood. And if it helps water reabsorption, then it decreases water content of urine, or the water content of urine decreases water content of urine. So you will have a concentrated urine, and this makes sense because when you have a concentrated urine, then this is against or anti-diuresis. Okay. By having finished the collecting ducts, we would have finished all the physiology and anatomy of the urinary system. I mean, we would have finished all normal structure and function of the urinary system. And now we're going to discuss the disorders of the urinary system, the clinical part that is always interesting to you and to all students, actually. The first disorder we are going to talk about, actually, it's not a disease, but it is a reflection of a disease, or it is an indication that there is a disease, and it is dysuria. Actually, dysuria is a symptom, and as you know, in biology and in medicine, the prefix dys means difficult. So dysuria is difficult micturition or difficult urination. Actually, the word micturition is the scientific word for urination. So difficult, where did I get that word from? This. So whenever, for example, dysarthria means difficult articulation of the words. Dysuria means difficult urination or micturition. Dyspnea means difficult breathing. So dyspnea, or sorry, dysuria means difficult micturition. Now, what kind of difficulty is it? It is painful micturition. So the difficulty here, let's make sure we are recording. Yes. The difficulty here is in the form of pain. So it is painful micturition. And that pain is sometimes interpreted by certain patients as being a burning sensation. So the patient feels an intense feeling or very strong feeling of burning during micturition. Usually when the patient feels this, it is an indication of cystitis. As you know, itis means inflammation. That's to say, as we said before, redness, swelling, and tenderness if we're talking about the signs, or pain if we're talking about the symptoms. And the prefix cyst here refers to the bladder, because cyst, as you know, is a hollow sac that is filled with fluid. So usually the patient feels burning, a burning sensation during micturition or during urination, and usually it is associated with a very small volume of urine, although the patient has frequency, meaning that when we say frequency, this is the noun and this is the adverb. So frequency is a symptom, meaning that every now and then the patient has an intense and very strong desire to void and to go to the toilet and to micturate. However, the patient gives very small volumes of urine, and actually we can even say, to show how small the volume is, we can even say drops of urine. So you feel that the bladder is full, you feel an intense desire to micturate, however, you give minimal drops of urine. And in addition to this, you have this feeling every now and then. Okay, so if you go tell this history to your medical doctor, he would think first place of cystitis. Dysuria is the symptom. Cystitis is the cause of dysuria. Cystitis, or inflammation of the urinary bladder, is the disease that caused this dysuria. So what would the doctor do in order to confirm the diagnosis? Diagnosis means that you decide which disorder this is. Diagnosis means that you decide which disorder the patient is talking about or complaining of or suffering from. And in order to decide which disorder it is, in order to diagnose, you have to do several steps. The first step is to listen to your patient, meaning that you would take the history from the patient and then you would examine your patient. Sometimes in cystitis, there is nothing weird during abdominal and external abdominal and pelvic examination of the patient. So you need to do certain investigations in order to confirm the diagnosis. Why am I saying to confirm? Because every step tapers the diagnosis. If you have several possibilities, if you have a differential diagnosis, meaning that you have in your mind 3 or 4 possible diseases that can cause this symptom, then you need to taper the diagnosis by examining the patient and then by doing investigations. So the most important investigation here is urine analysis. And at the end of the presentation, we would be discussing how a systematic urine analysis is being carried out in the lab in order to reach a diagnosis. So if you do a urine analysis to the patient, this urine analysis will reveal bacteriuria. It's not normal to find bacteria in urine. But if you find bacteria in urine, then this is highly indicative of a bacterial infection of the urinary bladder. And in order to detect bacteria in urine, you take a drop of the urine, spread it on a microscopic slide, and then you put a cover on it and examine it. And sometimes there are other instances where we stain the bacteria, but if we are examining the bacteria themselves, so this needs microscopic examination of urine. So it's not chemical examination, it's not physical examination, but microscopic examination that shows you the bacteria and that confirms the diagnosis. So if I ever ask you about the steps of diagnosis, you would say history, examination, and investigation. Through the history, you have several possibilities in your mind to interpret or explain the symptoms of the patient. through examination, you taper or narrow those possibilities or this differential diagnosis, and then through investigations, you confirm your final diagnosis. Sometimes, in addition to the presence of bacteria in urine, you will find pus cells in urine. Is it related? Yes, because when you have bacteria in urine, you would have white blood cells to fight those bacteria and to kill those bacteria. But after having fought the bacteria, the white blood cells die, and when they die, they form pus cells, which are the dead white blood cells that do not have nuclei. And if there is significant pyuria means pus in urine. And if there is significant pyuria, meaning that if you have more than 5 pus cells per high power field, HPF stands for high power field, means when you look through the high power lens of the microscope, you see a circular field. What you count the number of pus cells in that field. For example, this is what we see in the field, the high power field under the microscope. So you would say you would see, for example, bacteria, and then you would see the pus cells. If you see more than 5 cells per high power field, and if you see more than, for example, 100 bacteria per high power field, then this is definitely dysuria. So pus cells, as we said, are dead white blood cells. Another important symptom is polyuria. So the second symptom is polyuria, and as the name indicates, poly means many, and polyuria or much, many or much, both work according to what you are talking about. So polyuria means that you are producing much urine per unit time. So you produce a large volume of urine per unit time. So it is increased urine volume. So in this case, of course, there will be frequency, just like you had in cystitis, but it will be frequency plus a large urine volume. In cystitis, in the previous slide, we had frequency, but only drops of urine being excreted by the human body. So why would a patient suffer or a person suffer from polyuria? We cannot say absolutely or generally why would a patient suffer from polyuria, because sometimes polyuria is a physiological finding, meaning that it's not indicative of a disease, just like when you drink plenty of water. So polyuria means increased urine volume, and this increased urine volume may be physiological or normal. When I say normal, I mean a normal response of the body. A normal response of the body, meaning that you drink plenty of water over a short period of time, and this results in your body excreting a large volume of diluted urine. In such a case, of course, urine would be extremely diluted. But it might also be a pathological condition indicative of a disease, and we have 3 main causes for pathological polyuria. The first cause of pathological polyuria is renal failure. The early stages, because we will be discussing 3 stages of renal failure: early renal failure, mid-renal failure, and late renal failure. What is renal failure? When both kidneys are not functioning well. Actually, renal failure is when both kidneys stop functioning. But when we talk about early stages of renal failure, we haven't yet reached the state or the status of the kidneys being unable to make urine at all. But early stages of renal failure is the status where the kidneys are not functioning well. And specifically, they are unable to concentrate urine. So the main defect in early stages of renal failure is the inability to concentrate urine. So if I am unable to concentrate urine, then my urine will be diluted, okay? But more commonly, we have diabetes mellitus. Of course, you might have heard, or most probably you have heard of diabetes mellitus, and diabetes mellitus is due to deficiency of a particular hormone called insulin. How is it related? In diabetes mellitus, how is this related to polyuria? You have low insulin secretion by the pancreas, as you know. So you have low insulin secretion by the pancreas. This results in what? In high glucose levels in blood. High glucose levels in blood. And when glucose levels are high in the blood, then not 100% of the filtered glucose at the kidneys will be reabsorbed, because the amount filtered is huge. Not 100% of the filtered glucose is reabsorbed. Not 100% of the filtered glucose is reabsorbed. Why? Because this filtered glucose is huge. So some glucose is lost in urine. Some glucose is lost in urine, which we call glucosuria. Some glucose is lost in urine, and this is a condition called glucosuria, and this results in polyuria. Why? Because this glucose that is lost in urine is osmotically active. It withdraws water. So we have glucosuria or glucose in urine, and glucose attracts water. Glucose attracts water. So when you lose glucose in urine, you lose water, and this results in increased volume of urine. Of course, it would be diluted urine. This fact that glucose attracts water makes us say that, or allows us to say that, glucose is osmotically active. Glucose is osmotically active, meaning that it is capable of attracting water. So, of course, we'll be discussing this during the last part of this PowerPoint presentation, next session, inshallah, when we are going to talk about the pathogenesis and the complications of diabetes mellitus. The most frequent cause of polyuria amongst these three is diabetes mellitus, but we also have another diabetes that is very rare, and that is called diabetes insipidus. Diabetes insipidus is also due to a deficiency in a hormone, but it's not due to a deficiency in insulin hormone. It is due to a deficiency in antidiuretic hormones. So the patients who have diabetes insipidus, and that's a very rare disease, have antidiuretic hormone deficiency. Since they don't have antidiuretic hormone, which concentrates urine, then they will have diluted urine, and this urine will actually be very, very diluted. Of course, you know that antidiuretic hormone works mainly on the collecting ducts, but it also works to a certain extent on the distal convoluted tubules. The opposite of polyuria is oliguria, and oligo means oligo means few. And here we say oliguria with a U, which means producing a very small volume of urine, which is usually concentrated urine. Again, and just like we did in polyuria, we have two main causes for oliguria. We have a physiological cause that is not related to diseases, and we have a pathological cause that, or we have pathological causes that are indicative of diseases, that are indicative of diseases. And this physiological oliguria might be due, for example, due to decreased water intake. You haven't ingested water for long, or prolonged starvation. You don't find food, you don't find water for a reason or another. For example, during wars, during any catastrophic events, natural disasters of any kind. So people do not find potable, pure water, or potable water. Actually, potable means that is suitable for drinking, just like edible is suitable for eating. We have potable water, but it might also be indicative of a pathology or a disease. And as we said, we have the intermediate or the mid stages of renal failure. So in renal failure, we have early stages, late stages, and mid stages. So here, renal failure. Of course, when you reach the, and this is the proper definition of renal failure, the both kidneys stop functioning. So we usually define renal failure by describing the late stages of renal failure, saying that both kidneys, and it has to be both kidneys, because one kidney, if one kidney only is damaged, the other kidney can take over and do the function of both kidneys. So both kidneys will stop functioning. And if both kidneys stop functioning, then there will be no urine formation at all. And this is the next and last slide we will be discussing, which is anuria. We will be discussing today. Anuria. Anuria means there is no urine formation at all. And of course, this leads to accumulation. This would lead to accumulation of toxins. And if you have accumulation of toxins, then the cells would not function properly and you have death. But in the mid stages of renal failure, you will have oliguria. So late stages, you have anuria, no urine formation at all. Mid stages, you have oliguria, very concentrated urine, very small volumes of urine. And in early stages of renal failure, you have polyuria, you have large volumes of urine. And as I said before, so polyuria essentially is large volumes of urine. But when you have large volumes of urine, this is usually diluted urine. In oliguria, you have small volumes of urine and usually concentrated urine. In polyuria, when you had diluted urine, it was due to, since it is diluted, then it was due to failure of the kidneys to concentrate urine. But in the mid stages, since you have concentrated urine, then it would be the opposite. It would be failure to dilute urine. So it's very weird that when the kidney moves from, or when there is transition from the early stages of renal failure to the late stages of renal failure, the kidneys do the opposite. In the early stages, they cannot concentrate urine, and therefore they produce very diluted urine. Whereas when renal failure proceeds and becomes more advanced, there is failure to dilute urine, and therefore there will be concentration of urine. In the terminal stages, you would have anuria or no urine formation at all. And in such a case, we would need an artificial kidney. And there are, meaning that you would need a machine or a device that purifies or filters the blood. So here, dialysis means filtration, dialysis or separation of substances from blood. Dialysis means filtration or separation, or this is the closest meaning to dialysis. And you can take the whole blood outside the body of the patient, as we will see on the next slide, or you can do this filtration via the peritoneum. And we will be discussing both hemodialysis and peritoneal dialysis on the following slides. But you might also do surgery, and the patient might need renal transplantation, meaning that you implant a kidney inside the human body, a kidney that is functioning, of course, but each and every one of these has its own risks, its own dangers, and renal transplantation is referred to usually when hemodialysis or peritoneal dialysis are ineffective. Next time, inshallah, we will be discussing hemodialysis, peritoneal dialysis, and the rest of the renal symptoms. Thank you. And you will be receiving a worksheet for that is related to the content of this video. Thank you, guys. And you'll be receiving another video, inshallah, during the winter break, and that will be all for the winter break. Hope you will enjoy your winter break to the maximum, and see you, inshallah, after the winter break on Tuesday for grade 10A, and on Tuesday for 10B and Thursday for 10A. Bye.